Hi everyone, so today we're going to be talking about contact inhibition, NF2, and APC. So first we'll start off talking about what contact inhibition is. So usually when we, in a normal setting, when we culture cells, cells will form in layers, and cell-to-cell contacts will form between layers of the cells. So this contact between the cells will inhibit their proliferation. Cell-to-cell contacts in many tissues are mediated by interactions between transmembrane proteins called cadherins. So this mechanism, so we said normally this is what happens, but in malignant cells, in cancer cells, we lose this cell-to-cell contact and contact inhibition. And mechanisms that govern contact inhibition are just now being discovered. Between epithelial cells, E-cadherin is what mediates the cell-to-cell contact, and how this protein maintains normal contact inhibition isn't fully understood yet. One mechanism that sustains contact inhibition is mediated by NF2. So what is NF2? NF2 is a tumor suppressor gene found on chromosome 22. It encodes for neurofibromin 2, which is also referred to as Merlin or the schwannomin protein. So this Merlin protein facilitates E-cadherin mediated contact inhibition. If we lose NF2, we end up with something called neurofibromatosis type 2. And in neurofibromatosis type 2, we see things like meningiomas, bilateral vestibular schwannomas, and juvenile cataracts. So a loss of NF2 gene will lead to neurofibromatosis type 2. Another mechanism of E-cadherin regulation is the adenomatous polyposis coli gene, which is also another tumor suppressor gene. So far we talked about two tumor suppressor genes, NF2 and the APC gene. So tumor suppressor genes in general, let's talk about what they are or what they do. Tumor suppressor genes are just as they sound. They are genes that suppress tumors from forming. So they can be thought of in two different groups. One is gatekeepers. Gatekeepers regulate the cell cycle, so they produce products, or their products rather, are things that will regulate the cell cycle or lead to growth inhibition by cell-to-cell contact, like we talked about earlier. And another group are called caretakers. These repair DNA damage. They maintain genomic integrity to prevent tumors from forming. Gene deletion or loss of function mutations of tumor suppressor genes will lead to cancer. So now let's talk a bit more about the APC gene. APC gene product is a cytoplasmic protein. The main function of this protein is to regulate intracellular levels of something called beta-catenin. Beta-catenin binds the cytoplasmic portion of E-cadherin, and it also translocates to the nucleus and activates cellular proliferation. There's something called the WNT signaling pathway. This is a pathway that regulates cellular proliferation, and this ties in to beta-catenin and the APC gene. So how does that happen? So WNT is a soluble protein that can induce cellular proliferation. It binds to its receptor on the cell surface and leads to a cascade of events and signals that will end up blocking the degradation of beta-catenin. So blocking the degradation of beta-catenin leads to beta-catenin being able to be found in greater numbers in the cytoplasm, moving to the nucleus, leading to transcriptional activation in conjunction with a molecule called TCF. Cells that aren't really dividing much, they're not exposed to WNT. So lack of WNT leads to a lack of signals that block the degradation. So if we have no signals that block degradation, we end up getting more degradation. And beta-catenin gets degraded by a destruction complex, of which APC is an integral part. And if we don't have any beta-catenin going to the nucleus and leading to transcription of certain factors that lead to cellular proliferation, we get lack of proliferation. So this process is seen in cells that aren't dividing much. Now, the APC gene, if we end up having a mutation or loss of the APC gene, we will not have a degradation complex. So, in essence, the cell will behave as if it's under a constant stimulation by the WNT pathway. The WNT pathway, as we said, leads to signals that prevent the degradation of beta-catenin. And the degradation of beta-catenin is done by a destruction complex of which APC is an integral part. Now, a lack of APC, so no APC, no destruction complex, increased beta-catenin, it would lead to the cell behaving as if it's under constant stimulation by the WNT pathway. So no degradation of beta-catenin. We get increased levels of beta-catenin, we get transcription of cyclin D1, MYC, twist, and slug. So cyclin D1 and the MYC are growth promoting genes. Twist and slug repress E-cadherin expression and reduce contact inhibition. So as we can tell, these two things will lead to cellular proliferation, lack of contact inhibition, so haphazard growth. So now that we talked about that, what can lead to no APC or mutant APC? We have a condition called familial adenomatous polyposis. It's due to mutations in the APC gene. So from its name, we can see that it's a familial condition. Patients will have a germline mutation of one allele and then end up having a sporadic loss of the other normal allele. And by the time they're in their 20s or teens, they're going to have hundreds to thousands of adenomatous polyps. And eventually, these polyps will undergo malignant transformation and become cancer. Hope that makes sense. Thank you for watching.